The cell cycle is an intricately regulated process that ensures accurate DNA replication and cell division, orchestrated by cyclins and cyclin-dependent kinases or CDKs. Among these, mitotic cyclins such as cyclin A and cyclin B are central regulators of mitotic entry and progression. Their precise modulation is essential for genomic stability, as any dysregulation can lead to aneuploidy and oncogenesis. Post-translational modifications, or PTMs, of mitotic cyclins play a fundamental role in dictating their stability, localization, interaction with CDKs, and timely degradation. PTMs such as phosphorylation, ubiquitination, sumoylation, and acetylation influence cyclin function dynamically, ensuring proper mitotic progression. This documentary delves into the intricate world of mitotic cyclins, uncovering the biochemical modifications that fine-tune their activity and the profound regulatory consequences of these modifications. Mitotic cyclins govern the transition from the G2 phase into mitosis and regulate critical mitotic events, including spindle assembly and chromosomal segregation. Cyclin A activates CDK1 and facilitates early mitotic events, while cyclin B accumulates in the cytoplasm and translocates to the nucleus to drive mitotic entry. The tight regulation of these cyclins ensures that mitosis proceeds without errors, as any misregulation can lead to catastrophic consequences, such as chromosome missegregation and tumorigenesis. Cyclins themselves lack enzymatic activity but act as regulatory subunits that modulate CDK specificity and activity. The timely synthesis and degradation of mitotic cyclins ensure unidirectional cell cycle progression. Cyclin degradation is mediated by the ubiquitin proteasome system, particularly via the anaphase-promoting complex/cyclosome, or APCC, which targets cyclins for destruction at the mitotic exit. PTMs of mitotic cyclins determine their binding affinities, subcellular localization, and degradation, underscoring their importance in cell cycle fidelity. Phosphorylation is one of the most well-characterized PTMs regulating mitotic cyclins. Specific serine and threonine residues on cyclins are phosphorylated by kinases such as CDKs, polo-like kinases or PLKs, and aurora kinases, altering their stability, function, and interactions. For instance, phosphorylation of cyclin B1 at serine 126 and 133 by CDK1 enhances its nuclear translocation, a crucial step for mitotic entry. Conversely, phosphorylation at different residues can inhibit cyclin activity or trigger its degradation. Experimental studies have shown that cyclin B1 phosphorylation at 309-161 is essential for its activation of CDK1. In Xenopus egg extracts, mutating this site results in a failure to initiate mitosis, highlighting the necessity of this modification. Similarly, Plk1-Mediated Phosphorylation of cyclin B1 at serine 133 enhances its nuclear import by disrupting cytoplasmic retention motifs. In cancer cells, aberrant phosphorylation patterns of mitotic cyclins are often observed, leading to uncontrolled proliferation and chromosomal instability. Ubiquitination is a major determinant of mitotic cyclin stability, marking them for degradation at precise cell cycle stages. The APCC and E3 ubiquitin ligase recognizes destruction motifs DBOX and KENBOX on mitotic cyclins, catalyzing their ubiquitination and subsequent degradation by the 26S proteasome. Cyclin A is degraded at prometaphase, while cyclin B degradation initiates at anaphase, ensuring proper mitotic exit. APC/C-mediated ubiquitination is tightly regulated through its co-activators, CDC20 and CDH1. CDC20 drives cyclin B ubiquitination, and experimental evidence from HeLa cells confirms that CDC20 depletion results in cyclin B stabilization and mitotic arrest. Similarly, APCC inhibition in cancer models leads to persistent mitotic cyclin activity, promoting oncogenic progression. The interplay between phosphorylation and ubiquitination is also crucial, as phosphorylation of cyclin A at serine 154 enhances its recognition by APCC. Sumoylation, the covalent attachment of small ubiquitin-like modifier or SUMO proteins, has emerged as a critical regulator of mitotic progression. Unlike ubiquitination, sumoylation generally stabilizes proteins rather than targeting them for degradation. Cyclin B1 Sumoylation at lysine 63 has been reported to enhance its mitotic function by preventing premature degradation. Studies using SUMO-deficient mutants in human cells have shown delayed mitotic exit and defects in chromosome segregation, highlighting the role of sumoylation in fine-tuning cyclin degradation. Additionally, SUMO-modified cyclin B1 is resistant to ubiquitin-mediated degradation, suggesting a competitive interplay between these two PTMs. In cancer cells, altered sumoylation levels can contribute to mitotic errors and aneuploidy, further solidifying its role in mitotic fidelity. Acetylation of mitotic cyclins, though less studied, plays a role in modulating their interaction with CDKs and regulatory proteins. Acetylation of lysine residues in cyclin A, mediated by acetyltransferases such as P300, has been reported to enhance its interaction with CDK2, thereby promoting mitotic entry. Conversely, deacetylation by histone deacetylases or HDACs can suppress cyclin activity. Other PTMs such as methylation and glycosylation have also been suggested to regulate mitotic cyclins, although their precise roles remain to be elucidated. Recent mass spectrometry-based proteomic analyses have identified novel PTM sites on cyclins, indicating a more complex regulatory landscape than previously understood. Mitotic cyclins are subject to a complex interplay of PTMs that dynamically regulate their function. Phosphorylation can enhance ubiquitination, as seen with CDK1-mediated phosphorylation of cyclin B1, which primes it for APCC recognition. Similarly, sumoylation and ubiquitination can compete, affecting cyclin stability. Mathematical modeling of PTM interactions has provided insights into how these modifications collectively regulate mitotic progression. Computational simulations suggest that an imbalance in PTM dynamics can lead to mitotic errors, supporting experimental observations in cancer cells. Such studies highlight the potential for targeting PTM-regulating enzymes as therapeutic strategies in cancer treatment. Given their central role in mitotic regulation, mitotic cyclins and their PTMs are attractive targets for cancer therapy. Small molecule inhibitors targeting cyclin-CDK complexes such as palbociclib and ribociclib have shown promise in breast cancer treatment. Additionally, modulating PTMs using specific inhibitors, such as Sumoylation inhibitors, TAC981, and ubiquitinating enzyme inhibitors, is emerging as a novel therapeutic strategy. Studies in leukemia have demonstrated that inhibiting Plk1, which phosphorylates mitotic cyclins, can induce mitotic catastrophe and apoptosis in cancer cells. Similarly, HDAC inhibitors that prevent cyclin deacetylation are being explored in clinical trials. Targeting PTMs provides a promising avenue for selectively disrupting cancer cell proliferation while sparing normal cells. Post-translational modifications of mitotic cyclins are fundamental to ensuring proper mitotic progression and genomic stability. Phosphorylation, ubiquitination, sumoylation, and acetylation dynamically regulate cyclin stability, localization, and function, orchestrating mitotic events with remarkable precision. Dysregulation of these modifications is implicated in various cancers, highlighting their clinical significance. Understanding the interplay between different PTMs offers insights into targeted therapeutic strategies, providing a potential path for developing novel cancer treatments. Future research will continue to unravel the complexity of mitotic cyclin regulation, further refining our knowledge of cell cycle control and disease intervention.